Okay, so I just wanted to do a review for the lab practical in case you needed reminders or you missed class when I was talking about how to study. So you should have gotten your labs back that were graded and so that you could study for the lab practical. So remember, I'm just going to give you some examples of questions and stuff that you need to study like I did in class. And so remember that you are going to have to be able to recognize a cell that's in mitosis for the animal cell and the plant cell. So the first 8 questions of the test, that's what it's going to be, is remember we have prophase, metaphase, anaphase, and telophase for the animal cell, and then prophase, metaphase, anaphase, telophase for the plant cell. And of course they'll be out of order. It'll already be set up for you so that the pointer of the microscope's pointing at it, and the question will ask you what stage of mitosis is shown in the animal cell at the pointer, and then you look in there and then you need to be able to tell me if it's prophase, metaphase, anaphase, or telophase. And those are fill in the blank because obviously that's the only questions that you have. And so here it's showing, hold on, I'll get it straight. It's showing, animal cell across the top, and then the plant cell ones across the bottom. So make sure that you can recognize those. And then also I might ask you about the cleavage furrow in the animal cell and identifying the cell plate in the plant cell. So that will most likely be on there. And then you do need to know how to be able to bring the letter E into focus. Remember, that's going to count for 20% of the grade. That's an important skill, is just knowing how to use the microscope. And so that, as much as we've been using the microscope and what you've learned, you should be able to find the letter E very easily. Okay, then also you need to know about the meiosis. Let me now with meiosis, remember I actually had here, let's see if I hold it up like this. I had some examples of with meiosis where I had, let's see, prophase, and I showed you that one, and then I showed you one that was at the end of meiosis 1, so like telophase 1 or prophase 2. So I showed you guys those. I had them set up as demonstrations when we did that lab. And oh, you probably can't see this one. And then this one here, this is showing telophase 2, and it shows 4 cells. So that's at the end of meiosis, because remember, meiosis produces 4 daughter cells. So I might ask about one of those and ask you. I think also I showed you guys a demo of this showing metaphase 1 and where it had the chromosomes lined up. So I may have those set up as well, where I ask you what phase of meiosis, what phase of meiosis, or I may ask you, does this represent meiosis or mitosis? Like for here for telophase 2, I might say, is this cell? This is at the end of cellular division. Is this showing us meiosis or mitosis? And you should know that it's meiosis just from looking at it. And then also one of the ways you can tell is that it's 4 daughter cells. There's 4 cells there. So whereas remember mitosis, there's only 2 daughter cells at the end. You also have to know the parts of the microscope. That's going to be fill in the blank, where you have to know like the ocular lens, the stage, all of those things that you had to identify when we were first learning about the microscope. So I'll have those. And then I'm also going to have the animal cell model and the plant cell model. So here's the animal cell model, and you're going to have to identify an organelle that's on the animal cell. And I'm just going to ask one. But you'll have to know, like, you have nucleus and endoplasmic reticulum, mitochondrion, let's see, up here, Golgi apparatus, centrioles in the centrosome, lysosomes and peroxisomes I don't really ask, because on the model it's just difficult sometimes to be able to tell. They look exactly the same, except for on the model they're a different color. So I normally don't ask them. Oh, and then remember, you have the nuclear envelope on the outside, and then in the middle of the nucleus is the nucleolus. Plant cell will be on there too, and it's basically the same structures as the animal cell. So but it's also gonna have chloroplasts. Let's see, here's chloroplast there too, and it has the nucleus, nucleolus, ER, Golgi, mitochondria. Oh, and then the central vacuole. That could be a question on there, central vacuole. Okay. And then besides just being able to identify a couple of organelles there on the models, you have to be able to tell me some functions of organelles. So not necessarily the ones that you're identifying on the model. It could be another organelle. So part of that lab exercise was being able to identify the organelles, and then also being able to tell what their functions are. So go over that as well. Make sure you know what the functions of the organelles are. And then you also had to know the difference between prokaryotic cells, eukaryotic cells. So I will have that on the test as well, where basically I have it set up and I just ask you, does this represent a prokaryotic cell or a eukaryotic cell? So you have to be able to tell me if that, by looking at it. So just remember that prokaryotic cells, they don't have a nucleus. They're going to be much smaller. That's going to be the bacteria cells that we looked at. And then we looked at kidney cells for the eukaryotic example. And remember, they're going to have a nucleus. I also asked just some regular questions. Remember, I talked about how, and by regular, what I mean is there's not a microscope or anything at the station. It's just going to be a question. And so with those kind of questions, you need to just study through the lab exercises and look at, the review questions or bolded words that are in the lab exercises. And so just go through and do that. And then make sure also you know how to read the ruler for the centimeters and the millimeters, because I'll ask something about that. And I do ask one question that was from the lab where we learned about cell components and whatnot. And I ask a question about the protein test, and where we learned about doing protein tests, starch tests, and stuff like that. So basically I give you a question where I set up this is the test and these are the results that we did. This is what we did. And so then this substance we're testing for, it could be a protein or a starch or whatever it is. And so we did the test, and then this was the result. So this substance is what, and that one, so it could be a starch, lipid, protein, sugar. And then I do have, remember we learned about in meiosis the chromosomes, and you had to know with the pop beads, you had to be able to tell this is the centrosome, these are the sister chromatids, this would be a homologous pair, these are non-sister chromatids. So I do ask about that as well. So let me know if you have any questions. Just remember that on the day of the test, the door will be locked. We're all going to start the test at the same time, and there's about a minute per station. And once the time is up, you have to go to the next station, and you don't get to go back to look at the questions or change any of your answers after, to do that. So we all turn in the test at the same time. So, but please let me know if you have any questions.